Welcome to Toxicology lesson. Today we will talk about mutagenicity test. It is well known that DNA consisting of nucleotide bases play a key role in genetics. DNA transmits the genetic information from one generation of cells to the next generation through self-replications. During the transmission, spontaneous mutation may occur. Therefore, it may alter the amino acid content of the protein synthesized. Spontaneous mutation and natural selection are the major means of evolution, but in recent decades, a number of toxicants have been found to be mutagenic to a variety of organisms. A number of human diseases are the results of defects of the DNA repair system. The seriousness of this matter, therefore, warrants the extensive investigation in various fields of mutagenesis. At present day, there are a large number of mutagenicity tests. In this video, we will talk about mutagenicity test, which utilizes microorganisms, insects, mammalian cells, and intact organisms such as mice. Microorganism test in vitro of mutagenicity test involves prokaryotic and eukaryotic microorganisms. The Ames test, developed by Bruce Ames, is a mutagenicity test system which utilizes prokaryotic microorganisms. The Ames test depends on the ability of mutagenic chemical to bring reverse mutation of a certain amino acid-dependent mutant to a certain amino acid-independent wild-type organism. In this test, mutants are incubated in a medium that contains insufficient certain amino acid. If the toxicant added into the culture is capable of inducing reverse mutation, then bacteria can grow in the medium. Different mutagens may affect a strain of bacteria but not the other. In the mutagenicity test, which utilizes the eukaryotic microorganisms such as Saccharomyces, the mutagenicity of the chemical is determined mainly by detection of reverse mutation and, at limited extent, forward mutation. Some mutagens are inactive before bioactivation, which usually occur in the liver. To examine this, the mutagen is injected into the host, usually prior to the introduction of the microorganisms. After a few hours elapse, the host is sacrificed. The microorganisms are then examined for manifestation of mutation. These assays have the advantage of incorporating the biotransformation of the toxicant in the host mammals, but at the same time have the drawback that the microorganisms can only be kept in the host in a relatively short period. The fruit fly Drosophila melanogaster is the most commonly used insect in the mutagenicity test. It has the advantage over microorganisms that it metabolizes toxicants in a manner that is similar to mammals. Furthermore, it is superior to mammals in two aspects, which are: it has shorter generation time, only 12 to 14 days, and it can be tested in sufficient numbers at much lower cost. Another in vitro system for mutagenicity test is using mammalian cell in culture. After perturbating mammalian cells with toxicant, Forward and reverse mutation may occur in cells. The mutants respond selectively to nutritional, biochemical, serologic, and drug-resistant growth manipulation. These mammal cell lines are generally deficient in metabolizing enzyme. Therefore, such enzyme systems are often added. The last system in mutagenicity test is the system which utilizes an intact animal. There are at least 2 mutagenicity tests in mice. The first test is mouse spot test. In mouse spot test, pregnant mice are treated with toxicant. Then the mutagenicity is determined by formation of clones of mutant cells that are responsible for the color of the fur. Although the test may yield a false positive result, it has not yet yielded a false negative result. The second test is specific locus test. It involves exposing non-mutant mice to toxicant and subsequently mating them to mutable recessive stocks. Mutant offsprings have altered phenotypes expressed in hair color, hair structure, eye color, ear length, and other traits. There is a positive relation between mutagens and carcinogens. The result of mutagenicity test is a useful information to ensure the safety of any chemical to human, especially products used by humans, such as pharmaceutical products. Thank you for the Faculty of Pharmacy, Universitas Gadjah Mada, and the program Kompetisi-Kampus Merdeka